Hi, everyone, and welcome to another TrialSiteNews discussion. I'm Brandon, your host, and today we'll be talking about the Texas Senate Committee on Health and Human Services hearing from Tuesday, May 14th. We're joined today by Dr. David Wiseman. Dr. Wiseman is a PhD bioscientist with a background in pharmacy, pharmacology, immunology, and experimental pathology. Since 1996, he's run his own medical product R&D consulting business, and since 2020, he has focused on COVID-19. He's made over 28 submissions on COVID-19 vaccines to government bodies, including the FDA, CDC, and NIH. And he was a contributor to TrialSiteNews and participated in the Senate Texas hearing, which was on Tuesday. Additionally, we're joined by Daniel O'Connor. Mr. O'Connor is the founder and CEO of TrialSiteNews and recently spoke at the rally to reclaim free speech on the steps of the U.S. Supreme Court. Gentlemen, thank you so much for joining us today.
Good day.
Dr. Wiseman, tell us about the overall Texas Senate hearing event on Tuesday. Specifically, what was the aim? Who were the attendees, and what were some of the important topics covered?
Okay, well, thank you for having me. I'm going to share my screen really quickly so that you can see a little bit more. Okay, so on the screen is what is called the committee charge. Okay, so the way this works, I'm informed, is that the Lieutenant Governor who is the President of the Senate, sets the agendas for the various subcommittees and issues them with charges as to the topics that the individual subcommittees need to address. And so this is the charge that was given to this Health and Human Services Committee to identify and recommend ways to address the growing impact of cancer on Texans by evaluating state investments in cancer prevention screening, including but not limited to CT, MRI, and PET scans. Study and make recommendations on funding adequacy for prevention efforts at the Cancer Prevention and Research Institute of Texas. And just to explain what that is briefly, that is a sort of I'll say an equivalent, if you will, of the National Cancer Institute that the state of Texas set up a number of years ago. They raised a bond issue. I think the number was $6 billion. Daniel will probably know the better number there. But the idea was to make Texas the sort of number one location for cancer research based on a number of excellent centers of excellence, such as MD Anderson. So this was the charge of the committee. The committee consisted of, I think, 8 or so senators. There's 31 senators, I think, in total in Texas. So this was a quarter of the entire Senate that was there. And testifying along me were representatives from the Texas Health and Human Services Department, from this CPRIT organization, as well as MD Anderson and myself.
So let's talk about some recent cancer statistics. An American Cancer Society annual report on cancer facts and trends states while cancer deaths are falling, new cases are ticking upwards. More of these new cases involve younger people. That ACS report showed that younger adults to be only age group in increase in overall cancer incidence between 1995 and 2000. The rate has risen by 1 to 2% each year during that time period. But cancer is occurring in more adults at younger ages, even before they turn 40 or 50, and sometimes even earlier in those cases. These are called early onset cancers, which are diagnosed in adults between ages 18 and 49. What are behind some of these trends?
Well, I mean, this is the million-dollar question, and on the slide here is a sort of write-up, if you will, from the Yale publication on those cancer facts and figures, and sort of on the background, what made it sort of more prominent was the announcement by the Princess of Wales recently about her cancer treatment. But I think it's got everyone stumped, and the key in that report that you reference is that the incidence of early onset cancers has been going on for a while. This seems to have predated the COVID era, but along that we have to look at the mortality. So the difference between incidence, that means new cases that are coming in, being diagnosed, but there are reports as shown at the bottom of the slide here on increasing cancer mortality in the COVID era. So to address the specific question, what's causing it? There are several hypotheses, and these have all been talked about quite often. Did COVID, for example, in the COVID era, did COVID result in delays in screening, diagnosis, and treatment? In terms of going back before COVID, in these incidence numbers, have there been changes in health-related behaviors in general, physical activity, cigarette smoking? One of the topics that was brought up by the MD Anderson representative was possible exposures to plastics may have changed, different kinds of plastics, and so on. And we really don't know. There's an article in the Harvard Gazette also saying, well, could this be a birth cohort effect, some exposure, environmental exposure as yet unidentified, that goes back years and years. And I think it's got everyone stumped. And our contribution to this discussion is to say, okay, well, in the COVID era, in terms of mortality, okay, we cannot exclude SARS-CoV-2 virus, and we cannot exclude the vaccines. And the goal of the presentation was to say, listen, guys, we have to talk about these things. This is serious stuff. We have to look at all possibilities, but we've got to do it responsibly and methodically, and with the appropriate amount of caution and alarm, but not over alarm. But we've got to get the job done. So that's the sort of thrust of where I was going, and along with that, some of the comments from the other people in the panel.
I wanted to chime in on something, Brandon, if I may.
Sure.
First of all, Dr. Wiseman, yes, the CPRIT, it was a $3 billion initial bond underwriting that actually paid to get that very important research arm going.
Okay.
And I want to emphasize it was a very prominent group that Dr. Wiseman participated with in this Senate event. So highly impressive. I wanted to ask you, if you look online, there's a lot of chatter about COVID-19 vaccines and cancer, even the term turbo cancer, which I hear online on X, the former Twitter. You hear this term turbo cancer thrown around all the time, like it's a reality. From our position at TrialSite, just tracking the research, we can say that on the one hand, the medical establishment does not recognize turbo cancer, and there's no recognition that COVID vaccines or even COVID may have anything to do with cancer. On the other hand, there are this upticks in cancers. Specifically, I think you have identified some uptick in neoplasms, and I wanted to get your perspective on that. What have you identified with malignant neoplasms? How are we seeing this manifest clinically? And are there any hypotheses in terms of specific mechanisms associated with COVID-19, the virus, or the mRNA vaccines?
Okay, so there's a lot to unpack there. So on the slide here, Daniel, is a list of case reports. And again, case reports are not causal, they're not proving that there's causality, but case reports are appearing in the peer-reviewed literature of various different types of cancers associated with the use of any one of the four major COVID vaccines. There are, of course, VAERS reports, which I'm going to show you here, on the left top part of the graph. If you look at the number of reports in VAERS, again, this is not proving causality. and you normalize it by year, the number of reports per year for COVID vaccines of cancers is between 18 and 33 times more than all other vaccines, all other years combined. So that's a startling number. To try and begin to process that number into its significance, CDC have a method which is called this PRR signal analysis. And on the right-hand side, this is the actual output from the CDC's own data that was disclosed under a FOIA request. And you can see in the left column, there's about 12, 13, 14, 15, something like that, cancers that were reported associated with the COVID-19 vaccines. And to determine whether or not that begins to have significance, the CDC did this type of analysis, and very simply in the green box, if the number is more than 4, and in the red box, if the number is bigger than 2, that constitutes a signal. A signal doesn't prove causality, but it means we have to look at this closely. And this analysis was done July 2022, so coming up for 2 years almost. And to my knowledge, we haven't seen any further discussion of this in any of the FDA, CDC, or even the National Academies report recently about vaccine safety. So these are signals, and these are strong signals. And to say there's no evidence of an association is a problem. But to ask your question about the mechanisms, I'm trying to find a slide. Second. In terms of
that's slide 16. There you go. Yeah.
Yeah. Well, actually, let's just we cannot. First of all, we have to, we have to look at all possibilities here, Daniel. Okay, and there are possibilities relating to COVID virus, the SARS-CoV-2 virus itself, before we even talk about vaccines. And there are a number of papers, growing number of papers in the peer-reviewed literature, or even the preprint literature, suggesting that there are plausible mechanisms where the SARS-CoV-2 virus itself may do something that could influence cancer. And so I pulled out a paper from the NIH and NIAID. I pulled out a paper here from UT Houston and UT MD Anderson. I pulled out a paper from the National Cancer Institute and MIT, and lastly, paper from Brown and the University of Pittsburgh. Very credible workers. Dr. Elderi, one of the graduates from the Vogelstein lab. Very, very well-established cancer researchers. So there are a number of mechanisms that could contribute somehow to an association with the virus itself in cancer. And then when we translate that, when we go further than that and say, okay, what about the vaccines? Well, again, just to be clear, that the vaccines were never tested for cancer-related types of issues. And this is from the package insert from Comirnaty and from an FDA document. The carcinogenicity or genotoxicity types of studies were never done for these vaccines. So we don't have any baseline, and of course the absence of evidence is not the evidence of absence. But in terms of the vaccines themselves, what are the possible mechanisms? And on this slide, I listed a number. Of course, many of the mechanisms that could pertain to the virus itself, particularly relating to the spike protein, could apply equally to the vaccines, because the vaccines are intended to cause the body to produce spike protein. But there are a number of other mechanisms that are listed here that could be contributing to a possible relationship between the vaccines and cancer. And just lastly, to address the term that you mentioned, Daniel, turbo cancer. So it's become a sort of a popular term It's not. I don't think it's an official term in the medical literature. And I think we really have to. Um, we've got to do this right. And if this discussion becomes subject to sort of a political polarization for all sorts of reasons, then everyone loses. So we have to make sure we do this properly. We have to use the right terminology. We have to have the right amount of caution. But I don't think it's helpful to inject terms that are over-inflammatory.
I would agree, if I may interject here, that there are physicians out there with licenses that are pushing this claim very aggressively online. They're declaring that these are quote unquote turbo cancers, and that they imply that they're from the vaccines. It's in my opinion pretty reckless. On the other hand, I don't think we should, not, to your point, not look at this. Now, if we look at if the middle ground of doing some research on this to better understand what the signal is potentially coming from, could you share with us, again, we have a lot of researchers that come to TrialSite. Could you share what your priority would be if CPRIT gave you a budget to go look into this matter in the labs that you have access to in Texas? What would you prioritize, hypothetically?
Well, this slide, again, we're covering a lot of ground, and just to amplify that question, I mean, individual doctors who are seeing patients are reporting that they're seeing very strange cancers, and cancers that they don't normally see in a particular age group or with a particular presentation, and so on. So I can understand them wanting to give it a name, and I'm not sure how to handle that other than we've got to be careful in the use of our words. And of course, we're concerned that we're going to get a very large pushback from regulators. We already have, in terms of, this can't happen and it's impossible for all these reasons and so on and so forth. Whereas the truth of the matter is, the data is there, and I haven't shown you. I'll do so in a moment. The numbers, the statistical numbers showing the extreme statistical events that we're seeing in the numbers from the UK, from the US, our own analyses. So there's no question that something is happening, and it's not just early onset. It's in almost all ages. There's a very weird thing in 55 to 64-year-old age, but in pretty much all other age groups, there are very, very strong statistical signals for mortality, cancer mortality. Now, to answer your question, well, what do we want to do? So, I was sat next to literally the gentleman from MD Anderson, who I think was kind enough to point out about my presentation, that said, look, Dr. Wiseman was very careful in using words to use appropriate, sort of cautionary language. This is preliminary, and we need to investigate it, and it's like, but hypothesis, and so on and so forth. And he had no problem with that. And he was asked a question, or the panel was asked a question by the chairperson, Senator Colkhorst. Why is it so hard to talk about this? And he said and his answer, that MD Anderson, Dr. Hawk, he said it shouldn't be hard. It's okay to talk about hypotheses when it gets presented as fact. Okay, that's unsubstantiated or unconfirmed fact. Then that's a problem. And this is the tightrope that we're walking. And so I think the tone of the whole committee was very good. We had Democrats, we had Republicans. It was no sort of partisanship here. We had good comments and good questions from both sides of the aisle. And so to answer the charge of the committee, which was the purpose of us being there, I proposed or recommended that Texas invest in research to determine associations, if any, between cancers and the virus and the vaccines, and to develop appropriate methods of screening and so on. and that they fund these things adequately through CPRIT, which is their funding vehicle. And there's about 7 or 8 bullets here of a number of areas that we need to look at. We need to look at closely the statistics and to follow those. There was a really good question from I think one of the Democrat senators, Lamantia, who asked, in terms of the Texas Cancer Registry, are we capturing whether or not people had been vaccinated or exposed to or had COVID? And I mean, an absolutely brilliant question. And I mean, everyone agreed this is the sort of thing we need to be doing. So, again, this is not a partisan issue. It should not be a partisan issue. And you had a brilliant question there from one of the Democrat senators. So there's work to be done in terms of in the data. We've got our data set, which is what I presented, and we need to get the collaboration of the Texas HHS in order to really drill down on those data. That's one area. Second area would be the sort of more biological area, would be to get hold of cancer samples from MD Anderson potentially, as well as their library of genomic sequences, where we can, from cancers in the last 3 years or even going back before that as controls, where we can determine whether there have been events, integration type events occurring in the genome, either from the virus or from the vaccine. And we can, put that issue, to bed one way or the other. And the goal would be to develop screening methods, tools, services, products, which is one of the goals of the CPRIT, is to sort of with a commercialization type of goal, is to develop a service or product that could be related to assessing whether people's cancers may or may not have had a link to the virus or the vaccine. And then ultimately to see, well, are there specific things that we could do for those cancers if indeed the link is there? So, and then I put a plug in, of course, I know it's a subject dear and close to your heart, Daniel, with the React 19 clinic, which is going to be based in Texas, is that, we will have access to thousands of patients, not only from the React 19 vaccine group, but their partners, the Long COVID Foundation. I spoke to them also, and they were kind enough to let me use their name as clinical, samples, research samples that can help us get to that. So we've got a variety of lab-based things, of statistical-based things, of clinical-based things that we can look at. And there will be follow-up. I mean, that was very nice, that my colleagues on the panel were all very open to follow up. We've already began that process a little bit. But this was, I think, a historic moment, Daniel, where finally, I think we had a reasonable discussion, that wasn't partisan, that wasn't, polarized, for various reasons. And I think this is what science should be doing. So I was proud to be part of that moment.
We were really impressed that you got selected for that event, and having MD Anderson, which I believe is the top cancer center in the country, close to it.
Certainly.
Yeah, one of the top 3. CPRIT, which is the second largest funder of cancer research in the United States, next to the National Cancer Institute. Highly impressive. Let me ask you this: out of the group that was there, was there anybody that acknowledged the possibility? I know we have it up on the screen, and you mentioned it. Was there anybody else that acknowledged that there could be that this signal could be the result of either COVID-19 or the COVID mRNA vaccines? Was there anybody that acknowledged that directly?
Well, I mean, they acknowledged that it was a valid hypothesis. I mean, I think that's, I think, the comment from Dr. Hawk from MD Anderson was that, he had not heard until now of a possible association with these things, and he acknowledged this was a valid hypothesis, and he acknowledged that I was there to, I was cautionary in my language and saying, oh, this isn't definitely, I wasn't there. Oh, yes, that is definitely caused by A, B, and C. Acknowledge, the appropriate sort of language that I use and I always use. and he acknowledged that I was there to seek his help and the help of the other institutions. So I think that was that's all I could ask for, really. Frankly, I wasn't expecting that someone said, yes, you're definitely right. That wouldn't be appropriate, because I know we don't know the answer. So why would anyone else know the answer?
Right, right. Of course, I understand that, but I think the important thing is that it's been acknowledged, and that's critical because, to your point,
as an important hypothesis. Yeah,
yeah, as a hypothesis, but that's a big step because
yeah.
6 months ago you would have never had that happen. Okay, you and I know that.
Well,
Uh,
I don't think the forum has existed. I mean, you tell me, but I'm not aware that a forum has existed where you could actually have reasonable, I mean, it wasn't a dialogue because that wasn't the format of the meeting, but it was to an extent a dialogue, and the fact that everyone gave me their cards, they didn't like walk away and say, okay, don't ever call us again. That was not that wasn't the I got a nice email from one of the people, and so on. And I think the tone was really as it should be in science.
yes, most definitely. There is a hypothesis. There's a gentleman that has a YouTube channel who talks about IgG4
Right,
and how folks that get multiple boosts of mRNA
right.
tend to produce more of that. Could you maybe just explain, kind of simplify that for a broader audience? What
Okay,
does that mean?
so we have antibodies. Okay, I think everyone's heard of an antibody. They're these depicted these Y-shaped things that latch onto the enemy thing, like the virus or whatever, and somehow or other that initiates the whole chain of immune attack and destroys the enemy. So antibodies are these Y-shaped things, and there's different, and they're called immunoglobulins. So Ig is the abbreviation for immunoglobulin, and there's 5 major classes of them: IgG, AED, M, and G. Did I miss any out? Um, and they have various subtleties in how they work. And within those classes, there are subclasses. So the main class of antibody that we're normally talking about for this type of thing is the IgG antibody, but there are subclasses. And one of the subclasses is something called IgG4. Okay. And IgG4 is associated with the buildup of tolerance, immune tolerance. And it's exploited, in sort of, like allergists and so on, when you want to build up tolerance, for hay fever or whatever, that kind of thing. But IgG4 has been noted in a number of publications to have increased in its percent in people who have been vaccinated, frequently. And I think the number is about 20% in some cases, that it accounts for 20% of the IgG, which is a large number. And the IgG4 is basically involved in some level of immune suppression. So if you suppress the immune system, that means the immune system is sort of sent to sleep, or at least like they've had it, like they're driving under the influence, so to speak, and it's less able to combat infections or potential cancers. So the IgG4 increase is a potential mechanism whereby the cancer surveillance is reduced, and that could account partially for some of these observations that we're seeing in the COVID era.
Understood. Another topic I wanted to bring up, and Brandon, jump in if you like, but is related, and that we've been finding there's the frame shifting paper that came out in Nature. You've put out some intellectual work on that, and the whole topic of DNA fragments, in the vaccines, the mRNA vaccines. How much is still open to debate? There's different studies, but what's the connection to that? Because we hear that a lot about that, but I don't think a lot of people understand what that. It's complex what that is. Can you explain a little bit about how?
Okay.
Yeah.
Can I put my screen back?
Yes, please.
Okay. Well, let's talk about let's talk about frameshift proteins for a moment. So this is the slide I had. So this really sort of came to everyone's attention about December with this paper from the UK, Mulroney, from the group which is equivalent, or several groups that are equivalent of the sort of NIH, if you will, at University of Cambridge, Oxford, and other places. So a very, very credible group in Nature, obviously, very important, scientific journal. And they describe this phenomenon of frame shifting associated with this thing called N1-methyl pseudouridine. And that very long word is the thing, I'll just show it quickly, that won the Nobel Prize for Karika Wiseman in connection with these vaccines, this N1-methyl pseudouridine. What happens is that they discovered that if they substitute one of the gene letters in the gene code, the letter U, which stands for uridine, and they instead of using uridine, they use this methyl pseudouridine, a sort of a variant type of it, and they put that into the RNA. Then the RNA could evade the host defense mechanisms that are designed to look for foreign RNA, for example, from viruses, and so this is what enabled the vaccine, the Pfizer and the Moderna vaccines, to really to actually produce or evoke spike protein. So this was a major advance that won the Nobel Prize last year. But there's a number of potential effects of this, having this pseudouridine. One is it could involve the IgG4 pathway, as you mentioned a moment ago, and it could immune suppress in other ways, and it could result in this thing called frame shifting, which is again this paper here. And frame shifting means that if you have the genetic code, and instead of reading it from the beginning, you read it like shifted over a bit, you produce a different result. So the example here, the gene words are always 3 letters. So I just try to come up with a sort of an easy to understand example. Is I got 5 or 6 English words, A, Dan, T, Ton, Era, and said, okay, frame shifting means instead of reading the word from the A of the top word, you start reading from the second letter, the letter B, but you read them in words of 3. So now you can see that now the sentence reads, bed ant eat 1 rat. So the sort of sentence is a completely different sentence if you frame shift by just moving over 1 letter. So the pseudouridine renders the gene code subject to this kind of slippage, where the gene code is read, like 1 over, and when you read it 1 over, and when you shift it 1 over, it produces completely different sets of proteins than the intended one, the spike protein. And Mulroney showed that in patients, about 25% at least of patients were producing these spike proteins, these frameshift proteins, and they comment that this has the huge potential to be harmful, and it's essential that these vaccines are designed to be free from this problem. But then they sort of downplayed it. So, well, we didn't see any evidence of toxicity in our study. Well, yeah, you only looked at 21 patients, and these were patients that never had an adverse event. So, you really can't call that a safety study. But all the caution is there. So this is hugely problematic. The COVID-19 vaccines, the mRNA ones, this doesn't apply to the DNA vaccines, but the mRNA vaccines, right, are therefore producing unknown, uncharacterized proteins of unknown toxicology and distribution. I mean, we have no idea what they're doing. Okay, that's a huge problem. So that's the frameshift proteins. Now, as far as the DNA risk, DNA. Having DNA in the vaccines is a potential risk. It's widely acknowledged by FDA, by various guidelines, by Moderna patents, by things that BioNTech have written, that DNA that's in the vaccines could get into the genes somehow or other and play around with your genetic code somehow, and potentially, depending on where it ended up in your genes, could affect cancers by turning off your cancer suppression mechanisms or whatever. and this is widely acknowledging in international guidelines that go back, many years. Okay, the game changer here is that the guidelines were written when DNA that was in vaccines from chicken eggs and those sorts of things, it's there. No one disputes that it's there, but it's naked DNA, and its ability to get into the cell is minimal. But once you start on this slide putting the RNA and therefore the DNA into lipid nanoparticles, it changes everything. It changes everything because now your nucleic acids that are in your vaccines could get into cells tens or hundreds of times more efficiently than in the old days with the old vaccines, right? And so those guidelines really are meaningless for lots of reasons. And this is something that has not been taken into account and is being denied. But we go ahead.
Well, I was just going to interject that, the formal NIH CDC type of line is that the mRNA vaccines
Hello, I think you went on mute there, Daniel. Yeah.
We may have lost him. Yeah, I think what he was going to say is that their official line is that that does not. Is probably, I'm assuming, what Danny was going to say.
Right. So there was a letter in December from Surgeon General Florida, Dr. Ladapo, to the FDA and CDC about this, and can
you hear me?
We lost you for a little bit.
Okay, so what I was saying is that the formal established line with the mRNA vaccines is that they do not enter the nucleus of DNA at all whatsoever. Okay, right. And therefore they're not gene therapies. Okay, that's the official established line, truth. Yeah. Okay, so is that incorrect, Dr. Wiseman?
Yeah, well, I mean, there's lots to unpack. Before we even talk about these gene therapies, that's a separate discussion. Let's just talk about the response is that any connection between DNA and cancers is highly tenuous, because it is claimed that the DNA in the vaccines cannot get into the nucleus. Okay, well, that's not true. Clearly they can, and in the letter, the response to Dr. Ladapo's letter from FDA's Dr. Marks invoked that as one of the arguments, and what was interesting, he quoted a textbook, and I don't remember the name of the textbook, but he quoted a particular, very, very well-established textbook on cell biology, saying, well, look, the nucleus has this nuclear envelope which is designed to keep things away from the genes and so on and so forth. Okay, what he didn't realize was that in the same textbook that he quoted, like 2 or 3 chapters later, it talks about, well, what happens when the cell divides? When the cell divides, the nuclear membrane dissolves. Right? There's no nuclear membrane. So if you had DNA in the cytoplasm, meaning the non-nuclear part, during cell division, it has every bit access to the nucleus or to the genes. So in the same document, in the same textbook that FDA is citing, right there refutes their argument. Okay. Not only that, the spike protein and the SV40 sequences that we found can be nuclear localization signals, and so there's other ways that this stuff can get access to the genes, and there are other effects on cancers that don't even require access to the nucleus. And so there is a paper that came out. I'm trying to find it. Recently, here we go. This one here from He et al. That there's a concept of cytoplasmic DNA. It means that DNA that sits in the cytoplasm could actually do some stuff that could affect inflammation and cancers. So there are multiple mechanisms, right? Why we should be concerned about DNA in the vaccines. And again, what we found, particularly the work of Kevin McKernan, David Speaker, that I've been involved in somewhat, is that the amounts of DNA that we found are, depending on how you measure it, either approaching these guidelines even before we consider the lipid nanoparticle issue, or they exceed the guidelines by many-fold, right? When you measure the DNA in a different way, and this is another problem, is that even in the Moderna patent, they're acknowledging that there's different ways to measure DNA. And depending on how you do it, you're going to get different results. So clearly, when you measure it in a different way by fluorometry, you can get numbers in the dozens or low hundreds of fold in excess. And this has been confirmed by other people in various configurations. Dr. Buchholz in South Carolina, testifying in the South Carolina Senate several months ago, Dr. Sin Lee, and recently, and you wrote this, you had an article on this, I think yesterday, Daniel, on the work of Dr. Koenig and her lab, where they've independently found also increases in DNA by many hundred, or in their case, thousands of fold more. Again, we want to point out that we think that even though her work is confirming our work, her estimates of DNA are overestimates, because there's other quirks of how you measure it, which we're now learning. And so rather than say there are thousands of fold times more DNA in the vaccines, we don't think it's that high. It's still dozens or tens or maybe low hundreds, which is still hugely concerning, but it's not thousands of fold. And other people are confirming this. Preliminary evidence that there's integration in cell lines of the DNA from the vaccines, and the DNA may replicate. And once it replicates, then your guideline numbers are meaningless, because now DNA is being produced more and more, and it may mutate. But we need to confirm these results. We need to expand these and really get to the bottom of this. We can't ignore it. And so for FDA just to stick their head in the sand, I think it's just inviting trouble. They're entrenched. And again, in the presentation 2 days ago, I said, listen, we need to roll up our sleeves and do this work and do it formally, because the budget that we've had to do this, collectively, the different groups that are involved, the budget is zero. We've done this with zero budget. Okay.
Yes. Yeah. No,
right.
We understand that, Dr. Wiseman. You've been, leading the charge of in-kind
I'm not leading.
research.
I'm one of the people. I'm not.
Yeah.
I don't want to claim to be leading.
Leader of the movement, right? That's a joke.
Let's not go there. I'm certainly not the leader of anything.
It's a joke. But what I was going to tell you is, because we do got to wrap up pretty soon.
Yeah.
If we look at this, I would say significant milestone. This event, bringing together different scientists from different parts of the country in Texas, different persuasions, different political beliefs, what have you, all open to talk about these matters, listen to hypotheses. Want to again congratulate you on being accepted to present there. I think it's a really big deal, and again, we'll publish this and promote this fact in TrialSite. What's next steps for you and the folks in Texas and the various teams you're working with? Let's go back to the deck and just tell us what you're doing next. What can we look forward to?
So first of all, I do want to acknowledge, there are many people that are working on this, and, my current collaborators who are listed on this screen, as well as a number of people who've agreed to sort of, collaborate in various ways, on efforts going forward. And we've got a very distinguished panel here, Dr. Harvey Risch, a cancer epidemiologist, one of the foremost leaders in that field. Dr. Elderi, I mentioned him before, Brown University. Dr. Page, very prominent oncologist in North Texas with many clinics. The South Carolina Senator Dr. Senator Corbin, the React 19 COVID and COVID Foundation groups. So we're now sort of trying to say, okay, let's formalize what we want to do. Let's get funding for it. The number I asked for was $36 million. If CPRIT can fund some or all of that, if the other groups can at least provide data, genomic sequences, other types of non-financial assistance or collaboration, I think that's the way to go. There may be people who are willing to say, listen, this is a huge problem that's going to affect everyone. It will involve potentially, I hate to use the word commercialization, but I think some practical output where a product or a service, perhaps through a clinic, may come to fruition. But I think there's going to be some serious investors that I hope will look at this and say, we've got to look at this, and we've got to get this right. So I'm encouraged by the events of this week, but make sure that we pursue this with the appropriate tone, the appropriate caution, but with the appropriate diligence. And I'm thankful for the committee, the chair, Senator Colcourse, and the other senators for giving us this opportunity, and of course to all my collaborators that have done unbelievable work. So, and of course to TrialSiteNews, who've been at the front to bring these sorts of issues to the front and do it in a responsible journalistic manner.
Great discussion
thank you.
today.
Go look at TrialSiteNews. Subscribe today. Sign up. Use promotional code Wiseman.
Yes. Dr. David Wiseman and Daniel O'Connor, thank you so much for joining us today.
Thank you.
Thank you so much.
And for unbiased news and opinion articles and the latest in scientific research, please visit TrialSiteNews.com